going straight to the second speaker, Felix Drost. He's a PhD student at the Helmholtz Center in Munich at the Institute of Computational Biology, where he works on machine learning-based approaches to T-cell receptor epitope landscapes through prediction, multimodal integration, and representation learning. And today he's going to talk about mutant epitopes, which is very interesting.
Hey, do you see my screen? Good. Thanks for this kind introduction. As said, my name is Felix Drost and from the Helmholtz Center in Munich. So in this talk, I will focus, as said, on how mutations in the epitope can affect the T-cell receptor functionality. And because of this great talk before me from Peter, I can save almost my whole introduction. How we typically would assess specificity, of course, like the gold standard, even though it's noisy, is still the wet lab approach that we test a T-cell receptor or whole repertoire against the epitopes in combination with the MHC. However, of course, this is very very expensive, cost-wise, time-wise. So it would be quite cool to have computational models, as have been discussed in the previous talk, to take over this part. So I divided a little bit different than Peter. So one approach that is quite common is this database mapping. We heard about the databases, the IDB, the BDJDB, and so on. And so you could use your repertoire and query other identical sequences within these databases. And if there are, you can assign the specificity of these databases to your TCRs. So the issue here is in both approaches, in the wet lab approach and also the database query approach, is that you're restricted with regards to the epitopes that you can investigate. Typically, for example, if you have single cell multimer staining, you need to decide to a set of epitopes in advance that you want to test for. And of course, here in the databases, you can only query T-cell receptors for the specificity if the epitope is in the database. And that works quite well for well-researched diseases. For example, the IDB had been flooded recently with COVID epitopes. But of course, if you're in a regime where you have less TCRs annotated, it gets quite difficult. So this brings the field to machine learning-based predictors, as we have heard before, and especially like these multi-epitope models that can work on unseen epitopes. So as heard, we would insert the T-cell receptor, we would insert the epitope sequence and perform a binary prediction whether they bind or not. However, these methods are a little bit limited in that regard, often do not perform well for unseen epitopes until we get sufficiently enough data to train these models. So my talk, the model I will present here goes into this direction of machine learning-based prediction, but we don't present a generalized model that works on all epitopes, but we rather focus what happens if there are mutations within the epitope. And for that, we have a special data set where we had a deep mutational scan to the base epitope with the sequence single-amino. So basically what the team of Dirk Busch at the Technical University of Munich did, they took the single-amino epitope and mutated every position against all 19 other amino acids. And that led then to a total of 152 single amino acid mutations of this epitope, so 152 altered peptide ligands. And then we had T-cell receptor sequences, or T-cell receptors from two repertoires. The first one was from naive mice that had never been exposed to the antigen, but still filtered for the T-cell receptors that should recognize this base epitope. And second, we had 15 TCRs from an educated repertoire where the mice has been pre-exposed to the antigen. And also the model T-cell receptor for this epitope OT1. And then they did the wet lab work of actually testing the specificity or the reactivity of these T cells towards all these epitope mutations. And so you can see here the different T cell receptors on the y-axis. And you can see, which I find quite interesting, we have the educated repertoire with a much higher activation score and the naive repertoire with a much lower activation score. This activation score itself is used to be the percentage of activated T cells. However, we normalize this across the different receptors to have them comparable. And so therefore, it doesn't end at 100 anymore. So we don't call it a percentage of activated T cells anymore. What I find even more interesting is if you look at the x-axis. Here we have the different mutations grouped by the position at which the mutation occurred in the epitope. And the very interesting thing is that a single amino acid mutation can lead to a complete drop of the T cell activation. So if we look, for example, at position 4, we can see that here the T cells do not get activated anymore. And I think this is pretty remarkable. If you would imagine, for example, a virus that mutates at some point, and a single mutation could actually lead that an epitope does not get recognized anymore from a TCR repertoire that we have obtained by a prior vaccination or infection. And then from me coming from a machine learning background, of course, we ask, can we predict these effects? So we developed our model predicting T cell epitope activation by mutant versions. And we used this data set for that and splitted it. We considered every T cell receptor by itself, hold out a single mutation, and trained on the 151 remaining mutations. And did perform this then in a cross-validation setting and using random forests as a quite simple machine learning model, as the data set was not that big with 151 mutations. And as input data, we provided the epitope sequence, and where we represented each amino acid via a set of physiochemical properties, the so-called Edgeley factors. And this worked remarkably well. So especially for the educated repertoire and the OT1 receptor, we were able to achieve a performance of 90% in the AUC. And for the naive repertoire, we were also getting an AUC of a little bit about 80%. So, and the cool thing is we couldn't only do it in a classification setting for which we thresholded the activation score to represent in vivo recruitment, but we could also directly predict this activation score. So this is a little bit in line what was asked during the questions. For this data set, we had these normalized scores for all mutations. And so, as you can see here on the right side with the Spearman rank correlation coefficient, we were actually quite well able to predict as well this regression setting. Yeah. Of course, this model itself is not particularly useful. You need 151 mutations to predict the last mutation. That is not a setting that we want to have. So naturally, we tried to reduce the data that we need to train this model. And we tested the model by decreasing the amount of data we used for training. And we can see that for the educated repertoire here in orange and the OT1 in green, approximately 25% of the data set is enough to still have a comparable model. The naive repertoire, the performance dropped a little bit more when you're reducing the training data. However, it is very important that the sampling is uniform over all positions. So here in these 3 bar plot groups, you see on the left one, the base model, where we simply left out 1 mutation. So this is basically the most information the model can have during training. And the middle group is when we left out a position during training. And we can see here that the model doesn't generalize at all. So if we leave out a position when training our algorithm, the model cannot perform at all. However, when we leave out amino acid during training, as we've seen on the very right side, the model can still generalize because of this physiochemical representation we use when representing amino acids. So this was quite a good sign that our model actually made use of this representation. So here we saw we can reduce the amount of data to 25%. The question is, can we go even further? And we tried this by employing active learning. That is, we start with a small subset of labeled epitopes and train our model on this small subset. And we use the class probability of these random forests to estimate the uncertainty of all the other unlabeled epitopes. Then we would experimentally determine the specificity of the most uncertain of the epitopes where the model was the most uncertain, and add them to the training set and retrain the model. And then we have an iterative approach where at each step the model would choose which samples to label in order to increase the predictive performance best. Of course, we didn't do this iteratively with wet lab experiments, but we had the full data set, so we were able to simulate it by simply like withholding the label during training. And we compared this against random sampling of simply selecting random epitopes, and we could see that we at every iteration, at every step of training, we could actually reduce the numbers of samples we need to get a certain performance. So you can see here that we, with 24 APLs or like epitopes, we could reach a Spearman rank correlation coefficient of 70% and an AUC of 85%. And the interesting thing is here, we can also plot then, or like let the algorithm show us which samples selected at each step. So the initial sample at iteration 1, we uniformly distributed the samples over positions. So we had 8 samples where we had the largest distance to the base epitope measured by the Blossom distance for the sample. But then starting at the second iteration, we see that the model prefers specific positions when selecting new samples, which in this case, the position 8 and the position 3. So far, all the predictions were made within a TCR. So we didn't consider the relationship between the different TCRs. So the natural next step for us was, can we also generalize to a novel T-cell receptor? For that, we adapted the training split that we showed all the mutations, but held a complete T-cell receptor out of the training and then trained our models, also providing a representation of the TCR, which in this case, we used the CDR3 regions of the receptor. And again, we were able to achieve quite a good performance with an AUC greater than 90%, but mainly on OT1 and the educated repertoire. The performance for the naive repertoire was quite reduced at an AUC of 0.6. As said, there are many models around there. We also had heard previously about the IMREX model. So we tried to test the performance of these models on this data set. And we saw that these general purpose epitope TCR predictors do not capture these small changes that we had when introducing mutations to the epitope. This is, of course, to some degree an unfair comparison because our model has seen these different epitopes and these models have not. So our point was not that the model itself is better, but you need specialized methods to specialized data sets to estimate the effects these mutations have. What sparked then our interest is if you look at the naive repertoire, we have a quite diverse performance between the different TCRs. So we ask ourselves, why is this the case? So Here we see the performance for the naive receptors, and we saw that basically the performance correlates pretty well with the reactivity to the wild type epitope, to the base epitope. So we can see that we can generalize for novel TCRs if they have a high specificity towards the base epitope. And this also relates to the distance of the receptor to the remaining repertoire. So we measured the distance using the TCR dist algorithm and saw that the performance of our predictors correlates negatively with the distance to the repertoire. So we cannot, unfortunately, generalize to an arbitrary TCR, but if the TCR is reasonably similar and recognizes the base epitope to a high degree, then we can generalize to these TCRs. As a last step, we ask ourselves what drives the interaction. How does our model work internally? So for that, we performed feature important tests. We shuffled the input features and basically observed a drop in performance. So low performance means the feature is needed for the prediction and has therefore high importance. And our first test, we observed that when we assigned random TCR sequences to the model, the performance dropped most notably. So our model doesn't give an average, simple average of all TCRs, but actually incorporates the sequence with high importance into its prediction. But when we observe the importance of the features across all epitope positions, we could, for example, see that position 4 and 6 here have very high importance. So naturally, we ask ourselves what is going on here. So we resorted to structural modeling of the T-cell receptor. And then measured the distances between the epitope positions to its closest residue in the TCR, and observed that these position 4 and position 6, actually in very close proximity to the T-cell receptor, and therefore it makes sense that these positions influence binding to a large degree. Other positions, like positions 8, for example, that are reported in literature to be anchor positions, where the epitope is fixed within the MHC, showed quite low importance for the prediction, because naturally this is not a site where the TCR interacts with the epitope. So far, all these predictions, all the tests, all the experiments have been run on this one epitope with the sequence Synfigler. So naturally, we ask ourselves, does this also work on other epitopes? So I'm very thankful for the team of Dirk Busch, who then provided us 6 more T-cell receptors with a deep mutational scan against cancer-derived epitope. And so this time we had human TCRs. Again, we had quite a variety of different activation scores. But if you look now here at the position where the mutation occurred, we see that other positions are highlighted. So here, position 5 is very important for the T-cell receptor epitope interaction. If the mutation occurs, the activation will mainly drop. So it's hard to make generalized statement for the different epitopes, because this is, of course, majorly related to the MHC type. I will not replicate all the results here. We did this. If you want to see this, you can check out the preprint. But overall, we had a similar performance for all the tests as in the educated repertoire. So we saw that our model works reasonably well. What I want to highlight, however, was when we performed the active learning part. We also saw this, that we can generalize for 24 epitopes only. We've reached quite a high performance. But the model itself chooses different epitopes. So, of course, in the iteration 1, by default, we design our algorithm to give one mutation at each position. But then after iteration 2, when the model actually choose its samples by itself, we observed that it focuses on other positions, which is here position 1, position 3, position 4. So this gave us the insight that actually kind of this active learning approach is needed, as we don't know a priori which APLs or which epitopes we should test or we should experimentally measure. But this is better if we let the model decide this by itself. So this brings me already to the end. So I want to highlight the effect of mutations in the epitope can be predicted, and either within one T-cell receptor or across T-cell receptors that are related to the base epitope. And we cannot only treat this as a discrete binary event of activation or not activation, of binding or not binding, but if we have the correct annotation, we can also predict these continuous reactivity levels. However, this of course needs a data set where this is annotated. Of course, I want to thank everybody involved in this project here at the Helmholtz Munich. That's mainly the research group of Benjamin Schubert and at the LMU from Bernd Bischel, who these two groups did most of the computational work. But I want to give special thanks to the research groups of Kilian Schober and Dirk Busch for providing us this cool data. As we've heard, the data is basically the bottleneck for most of these predictions. So it was very good, very pleasant to have this good annotated data. If you're now interested, I think there's some time for questions. You can also reach out over Twitter or contact me by mail. Yeah, thanks for your attention.
Thanks, Felix. Super nice talk. Questions? So maybe I start with, so I really like the deep mutational scanning data. I think this is exactly what we need. So now you just looked at single mutations. Are you guys also working on multiple, 2 mutations, 3 mutations?
We have not so far. That's on our to-do list. But I mean, the issue is that then it gets hard what to test. Because like it grows basically quite drastically the amount of epitopes you could form. We had the idea to extend this active learning framework that we could basically test the mutations that will most likely provide us with good predictors if we introduce novel mutations, but we haven't started that yet.
And for the TCRs, those are more different from one another, right? Or they are also mutants?
Yeah. They were selected basically from repertoire data.
And did you see a trend that I don't know, that you could predict well the binding of more similar than of more different TCRs? Did you see some similarity there? And I mean, your prediction was quite high already, but if you
No, definitely. I think I showed this earlier. Here on the right side. So basically, the more distant the TCR is from the remaining repertoire, the lower is the performance.
Okay. And are you guys also planning on mutating some TCRs also to do some the similar things that you did on the epitope side, also on the TCR side?
As far as I know, this is not planned. It would be very interesting to have this data. I guess it's harder to produce. So the TCR were re-expressed in Chukat triple parameter reporter cells. And I think that would be very costly and time-consuming to have a very deep mutational scan on the TCR side.
Interesting. Questions from others? No questions? For the I have a question for the structural modeling. Was this a real QIUM structure, or did you just no?
We predicted the structure based on a model which is called TCRP MHC models. So like a structural model prediction tool specifically for TCRs and epitope MHC.
And that works well? And that works well? So because what we see in the antibody field, that there are many different structure prediction models, tools, many more than for the TCR world. And what one sees is that some models are better than others, and some are more accurate for some specific things. So some side chains are, it's better than others. Yeah.
I cannot really state how accurate it is. Like it was definitely good to see these positional distances to the epitope. That was captured quite well and was in line what have been previously reported in literature. However, we tried also to predict based on these distances and were not able to reach that high performance.
And did you try to just replicate it on known structures where we know what the structure is, and then try to see if the model replicates those known structures?
I have not tested the model in detail.
That would be interesting.
Unfortunately, also none of our TCRs and TCR epitope combinations that we had was like, for none of them there existed a structural model like a real one. That was quite unfortunate.
Yes, we have the same problem in the antibody field. Any more questions for Felix? So I just ask more questions then. So this is now one matrix. Is now the plan to make many of these matrices for many epitopes to get a more, maybe in the future, if one then combines all these matrices together to get a more generalizable model at some point of TCR epitope binding?
Ideally, I would want to have this, yes. So where we have this here for the synfetal epitope and one for one human cancer epitope, we are currently working on a third one. However, this is of course simply a lot of work in the wet lab, and my collaborators are not too happy to scale this to 20, 30 matrices. And I mean, we had this prediction earlier that maybe 5,000 epitopes would be needed for generalized models. I think that would require some group effort to really deeply characterize many deep mutational scans. And I think that would be super valuable for training these generalized predictors as well. As we have seen, they do not particularly well on mutation data. However, I think this is quite a laborious adventure.
Great. Then I think we're at the end, if there are no more questions. Thanks, Felix, again for the very nice talk. And thanks everyone for being here today. We are back with another AIRR-C seminar in a month. This one will be posted on the YouTube channel in 1 or 2 weeks. Thanks everyone. Bye-bye. Bye-bye. Thanks, Felix. Thanks, Peter.
Bye. Thanks.
Bye.